Recording in progress.
Wait a minute. Okay, now we will be discussing the formation of membrane-bounded organelles. The how different organelles are formed from the phospholipid. Like in this section, we will discuss what is the involvement of phospholipid in formation of membrane-bounded organelles. It is quite simple. As I told you that all the organelles, if they have membrane, their membrane will be formed by the phospholipid bilayer. Okay, every membrane in the animal cell, it will be formed from the phospholipid bilayer. So it is mentioned over here, the formation of membrane-bounded organelles from phospholipid involves these four steps. What are these four steps? Three steps actually. First one is the synthesis of membrane components. Let's talk about lysosome. Like we have lysosome. This is a membrane-bounded organelle. So how its membrane will form? How its outer membrane will form? Firstly, there will be synthesis of the membrane components. What are the membrane components? The components which will be present in its membrane. And what are those components? There will be phospholipid bilayer, there will be cholesterol, there will be proteins. Okay, these are the general things which will be present in almost every phospholipid bilayer in every cell membrane. Are you getting this? What I am saying? I am saying, firstly, when we have taken the example of lysosome, if you have lysosome and you are talking about how its membrane will form, so in the first step, when the membrane formation is happening, in the first step, the components will be synthesized. The components of the membrane will be synthesized.
Yeah,
And what are those components? What can be in the cell membrane?
the phosphate group,
The phosphate group.
the acid.
There can be.
There,
No, I'm not just talking about firstly, the main thing is phospholipid bilayer. That will be always there. And in phospholipid bilayer, these are the three things. What are those? The phosphate group
the fatty acid chain,
and
and the glycerol.
glycerol. Yes, glycerol. I'm talking about the other component as well. In the cell membrane, there is not just the case that you have just phospholipid. There will be the other components as well. There will be proteins as well. There will be in the cell membrane. There will be glycolipids, the conjugated molecules we have discussed in the chapter number 2. And there can be cholesterol as well. Okay, I'm talking about these components. So firstly, in the first step, when the membrane of lysosome is forming, what will be formed first? The components. The components will be synthesized. And what are these components? These are those components which will help to make the membrane of the lysosome. Clear? Now
Yeah,
these components will assemble and now they will form what? They will form a bilayer. The main in the bilayer, what will be the main component? The phospholipid. Okay. And in that phospholipid, you will have the protein embedded in it. The glycolipid will be present over it. The cholesterol molecule will be present on it. And then in the last, there will be remodeling through budding and fusion. What is meant by remodeling? That there will be, if let's suppose this is the membrane of the lysosome, there will be budding and there will be fusion. What is fusion? Maybe some part of this lysosome was formed over here, right? This part, and this part formed over this side, okay, at some space. So they will fuse together, okay? They will fuse together and they will form this whole one membrane. Are you getting this? What I'm saying?
so what is budding?
Budding is separating. Sometimes there is extra material, sometimes there is extra material, so that extra material will bud off. Okay, that is known as budding.
Okay,
So these are the two scenarios which will help to remodel. Okay, remodeling, there will be remodeling. So first step is the synthesis of what? The components. And what are those components? Firstly, mainly you have, can you recall
recall
the
what?
things in the cell membrane, the components of the cell membrane?
There's glycerol, fatty acid,
Firstly,
phosphate chain.
the most important thing is What? Phospholipid bilayer. Without that, you don't have any layer. Okay? You don't have any membrane.
By there.
Hmm. Then,
Um, then the proteins,
hmm.
the glycerol,
Yes,
fatty acid, and cholesterol.
yes. Cholesterol, glycolipids, glycoproteins, and proteins, transport proteins. These all things are the components of the cell membrane. Okay? So firstly, there will be synthesis of these components. Then what will happen? There will be assembly. Assembly of what? These bilayers. Initially, you have just one layer like this. This is one layer. So when after that, there will be assemblage of all the things. All the things will be assembled together. And in the last step, what will happen? Remodeling
Uh, Remodeling and
through budding and fusion.
budding.
The remodeling happens through
Yeah.
budding and fusion. There can be budding of the extra material and there can be fusion of the two things which needed to be present in the cell membrane. And this process is driven by the amphipathic nature of phospholipid which have hydrophilic head and hydrophobic tail. Now they're saying this assembly happens through amphipathic nature of phospholipid. Now, what does it mean? It means that the phospholipid has two types of inter. It can do two types of interaction. It has two interactions. From the outside, it will interact with the water, and from the inside, it will interact with the non-polar things. Okay. So, according to this, these molecules will arrange themselves. Are you getting this? Like the cholesterol is present over here, inside, okay? Inside
Yeah.
the fatty part. Transport protein is present like this, the extrinsic one. This is the intrinsic part. This is transport protein. Then you have the glycoprotein. These molecules, they are saying that these molecules will be arranged in the phospholipid bilayer according to the phospholipid bilayer's amphipathic nature. Okay, it is not just random arrangement. It is not the case. They are arranged according to the phospholipid nature. I told you that there is a concept of like dissolve like. There's also a concept of interaction between like things. Like it means obviously when you are interacting, if this is the case, if this is the carbohydrate, so this carbohydrate it is interacting with the hydrophilic part of this membrane, right? So it means this carbohydrate's polar part will be over here. Are you getting my point? I'm
Yeah.
saying you have different things. You have these different things, the glycolipids, glycoproteins, the cholesterol protein. These all things are present in the phospholipid bilayer. Now we are talking about how they are arranged. They are arranged according to the amphipathic nature of the phospholipid. Like if we talk about the cholesterol has far more greater interaction with the fatty part. So it will be having more space, it will cover more space in the fatty part, in the hydrophobic part. Are you getting this?
Yeah.
And the protein, some proteins, they tend to move, they tend to change their position from one place to another. So these proteins will be present like this. Clear?
The proteins can change their position.
Yeah, they can change. Some proteins. Some protein can change their position.
Okay.
Not all. Now this is mentioned over here. That this process is driven by the amphipathic nature of phospholipid, which have hydrophilic water attracting heads and hydrophobic water repelling tails. Are you clear with all these things?
Yeah.
And this is mentioned over here, the compartmentalization, which I was talking about. That you have membrane bound compartments. This is a membrane. Inside compartment, there is an inner compartment, there is an outer compartment, right? So how it is interacting with the inner compartment and outer compartment, it depends. What is in the inner compartment and what is in the outer compartment. Okay, just like this, I
Okay.
told you. Like if there is aqueous environment the within the cell, so this will be interaction. Okay, and if there is aqueous environment outside, like over here, so the mycel formation will be like this. Always remember this thing that hydrophilic head will always interact with the aqueous part. Okay, with the hydrophilic head of the phospholipid will always interact with the aqueous part, the polar part.
The hydro.
Hydrophilic part of the
Yeah.
phospholipid interact
Interact
with
with
the,
water.
yes, with the aqueous part or with the water. Simply, you can say. I can write over here. Hydrophilic part of phospholipid always interact with water or aqueous or polar part of the cell. Clear?
Yeah.
Okay. Now we have different things. Mm, Can you tell me this? How there is um, how the membrane of the organelle is formed their membrane by the help of phospholipid.
The formation of membrane bound organelles.
Mm-hmm.
The first is the synthesis of membrane compounds. The
Components.
proteins are, yeah, components.
Mm-hmm.
The proteins, the glycolipid, cholesterol, and phospholipid are formed, and they assemble into bilayers, then go through reformation, remodeling through
Hmm.
budding and fusion.
Yes. And what is budding?
Budding is the separation.
The bud of. It is. You will discuss it in bacteria. We will discuss in bacteria. Budding is like bud formation. You know the smaller buds. The extra material will bud off. When you have this assembly, the extra unimportant things will be bud off from the main organelle, main membrane of the organelle. And fusion will happen of the things which are important for the membrane. Okay, those things will fuse together.
Your
And
voice is kind of glitching.
hmm,
Your voice is glitching a little bit.
is it clear now? I don't know why it's happening.
Yeah, maybe it's the Wi-Fi.
Maybe. Okay, your voice is clear to me.
Okay.
How these components will assemble? How this process will
The,
happen? On which thing it depends?
the, amph- the amphipathic nature of phospholipids,
Hmm.
the hydrophobic and hydrophilic nature.
Yes, this is the main thing, the amphipathic nature of the phospholipid. It allows different components to arrange at different positions within the cell membrane. And this is the reason, the amphipathic nature, that some proteins can move and some proteins remain at their position. Okay. And then you have the fluid mosaic model. Now we will be discussing the model of the cell membrane in detail, the fluid mosaic model. And this was given by the two scientists, the Nicholson and the Singer in 1972. So it was the most acceptable, this is the accepted model for the cell membrane. Otherwise, there was a model, we have discussed it. What was the name of that model? The
For what?
lock and cell membrane, like the lock and there was not the, I'm confusing it, that was for enzymes. There was another model for cell membrane. There were many models, but this is the most accepted one. This is the accepted one. This model of the cell membrane, like in which they discuss that there are phospholipid bilayers. These phospholipid bilayers, and there are different proteins and different molecules arranged and present within the this bilayer at different positions. So at this, it describes the membrane as a fluid. Okay, it describes this membrane as a fluid. That is why the proteins can move. Some proteins can move because according to this model, what they're saying that this membrane is a fluid. Are you getting my
The
point?
what?
Fluid. This one.
Yeah.
So it describes the membrane as a fluid, flexible, and dynamic bilayer of phospholipid that act as a sea. So what they're saying,
Yeah.
the that
What is
the
this?
yeah, with various protein and other molecules floating within it forming a mosaic pattern. Simply what they're saying that what they suggested was this. They suggested that, like for example, this is a sea, okay, and inside the sea there are different things that are floating at different positions. Okay, what is happening? Like for example, you have like this is protein, this is a protein, okay. This can be a carbohydrate, clear? This can be a cholesterol molecule. Different things are present in the
Yeah.
phospholipid, okay. So they are saying this phospholipid bilayer is a C. This phospholipid bilayer is a C, and these all components, these all components are what they are doing. These components are floating in the
So they're
sea.
floating in.
In the sea, yeah. So this is what they're saying. And this the the phospholipid bilayer as it is acting as a sea. So they mentioned it as the fluid. They given it the name fluid, and the components they are floating. So these components are floating. That's why they gave it as the name of mosaic. Okay, that is why this is known as fluid mosaic model. I read this again. What they are saying. Can you read this?
Yeah.
Mhm. It
The
describes.
fluid mosaic. It describes the membrane as fluid, flexible, and dynamic bilayer of phospholipids that acts as a sea, with various proteins and other molecules floating within it, forming a mosaic pattern.
Are you getting this? What they are saying? That this
Yeah.
phospholipid bilayer, it is acting as a fluid. This phospholipid bilayer is acting as a fluid, and all the other things are floating in it. So all these components which are present other than the phospholipid in the cell membrane, all these components are acting as a mosaic. They are forming mosaic. Okay, because they are floating. This is the most accepted uh, model. Now, fluid mosaic. Fluid is what? The membrane is not rigid. What is meant by fluid? This I have written. Because sometimes they ask you in the short question in the P2 that what is meant by fluid and mosaic. So fluid is what? The membrane, it generally means the membrane is not rigid. If you we are not going to the literal meanings of the fluid. We are just saying that it means it allows different substances to pass through it. Okay? The membrane allows different substances to pass through it. The membrane is not rigid. Whenever someone asks you what is meant by fluid, like we are saying the cell membrane is a fluid mosaic model. So how in the membrane there is fluid? So you will say that this membrane is not rigid. Okay, this membrane will allow different substances to pass through it. That is why it is known as fluid. Clear?
Okay.
It is
Yeah.
not rigid. It behaves like a two-dimensional liquid. The molecules within it can move, okay? And laterally allowing the cell to change shape, repair itself and move. This is the reason. Okay, this is very important thing. This is the reason they have mentioned that because of this fluidity in the cell membrane, the cell can change its shape. Clear?
Yeah.
It can repair itself. It can move from one place to another just because of this fluidity in the cell membrane.
We can change its shape.
Yeah, it can change its shape as well. Because cell is, when we talk about the plants, we say that plant cell are rigid. their shape is not flexible. The shape is same, like there is a large vacuole in the center. But when you talk about the animal cells, they can change their shape. Yes, there is flexibility. And why there is flexibility? Because phospholipid act as a C. It is flexible in nature. The phospholipid allow different substances to pass through it. That is why the cell can change its shape only when it is needed, not always. When there will be need of changing the shape inside body, it will change its shape. We will discuss these things, inshallah, how this
Oh,
happen and why this happen. I'm just telling you that this happens. Sometimes cell changes its shape according to the need of the body. Clear?
yeah.
Now what is mosaic? The membrane is patchy mixed composition. Is a patchy mixed composition. You see in the membrane there are different things. It's not this. It's not like this. You cannot say like uh, you don't have a structure like this.
It's patchy, it's meaning it's not even.
Yes, it's not even. It has different things in it. Otherwise, if it was even, it means the structure will be like this. Clear? You must know how to draw this, okay? They ask in P2 in the long question as well. You must know what are the components, and this is very important. This is very, very important. And you must know the labeling as well. Sometimes they give you the question just to label in the P1 to label this. What is this? What is this? What is this? Okay. So mosaic is what? The membrane is patchy, mixed, there is mixed composition. It is composed of phospholipids, protein, cholesterol, and carbohydrate, which appear like tiles in the mosaic. Okay, like tiles in the mosaic when viewed from the above. If you view from the above, it will look like there are tiles in the. This is a mosaic pattern, and in this mosaic pattern, you have different tiles. Okay, if you see from the above, from the
Okay.
top. Clear?
Yeah.
Now, can you tell me what is fluid mosaic model?
It's a model that displays the cell membrane as a um, fluid and flexible sea
Mm-hmm.
with the with its components floating within it.
Mm-hmm.
It is fluid, so it can allow it's fluid, so it can allow the cell to change shape, repair itself, and move, and also let the other substances and even enter the cell. And
Yes.
it is mosaic, meaning the membrane is patchy
Mm.
and it appears as um it appears as tiles if you see from above.
Yes. It looks like the tiles in the mosaic when you view from the above. And
That's for the
according,
structure, right?
yeah, this is the structure. This is structure of the fluid mosaic model. This is not the structure of phospholipid. What is the structure of phospholipid? We have discussed earlier. What are phospholipids? This is phospholipid bilayer. Okay, this is phospholipid bilayer. Don't confuse. These are two different terminologies. This is membrane. You know, in membrane there is not just phospholipid. You have different things. Phospholipid is the main thing. Along with this, you have different components in the cell membrane. And what are those components? Like there are,
Phospholipid.
along with phospholipid, what is present in the cell membrane?
There's glycolipids,
Hmm.
there's proteins,
Yes.
there's cholesterol,
Yes.
and glycoproteins.
Yes. Exactly. These are the things. So I am saying don't confuse it. Phospholipid is just one component of the cell membrane. Okay. There are many. There are many things in the cell membrane. You can't say that there is just phospholipid, and you cannot. You also cannot say that you just have cholesterol, glycoprotein, and protein in the cell membrane. Without phospholipid, cell membrane cannot form in the animal cell, okay, or in the plant cell. Cell membrane is same. But the composition of these components will vary, okay? In different organisms, in different species, this composition will vary. Like in some, cholesterol will not be present, but in animals it is present. Clear? Are you clear with this? If you have any question, you can
Yeah.
ask. Like till
No,
this.
I don't have.
Okay. Now there is a Singer and Nicholson inside. What is their inside? They replaced the old sandwich model. Yeah, there was a sandwich model. And in that they just say that the proteins are sandwiched and there is lipid core, the inside core is lipid and outside there is protein, according to that model. What they said, that lipid is in the center, just like this, the lipid is in the center and proteins are outside, the whole, as a whole proteins are present outside. So this is what some people suggested in the previous times, some scientists suggested. So that model was rejected. And why it was rejected? Because no, proteins are not present in all throughout the membrane. They are not present throughout the membrane. They are present at different positions. Okay, and they can move and they are not rigid. According to that sandwich model, the proteins are the rigid. They form the rigid protein layer. That is not the case. Okay,
Okay.
and these proteins, now we will talk about the proteins which are present over here. These proteins can be of two types. These can be intrinsic proteins and extrinsic proteins. From the name indicated, intrinsic means they
Your
will
voice,
be, sorry,
your voice is glitching. Could you?
I don't know why it is happening. Why is it so?
Recording in progress.
What are those?
What are what?
Intrinsic and extrinsic protein. Can you tell me what comes in your mind from the name intrinsic and extrinsic?
Intrinsic means the proteins that are inside, and extrinsic means the proteins that are outside.
Hmm. The intrinsic proteins, we are talking about the cell membrane. We are not talking about the inside the cell. Okay, we're talking about the
The membrane.
membrane. So intrinsic proteins, they are embedded in the membrane with their arrangement determined by the hydrophilic and hydrophobic regions. Okay. Now their arrangement will depend on the hydrophilic and hydrophobic nature of the phospholipid bilayer. This we have discussed earlier, that the phospholipid bilayer's amphipathic nature decides what will be the arrangement of different substances within the cell membrane. Are you clear with this? If I ask you in the test that how amphipathicity of the cell membrane tells you the how the component will be arranged. So what will be your answer? How amphipathicity helps to tell us the arrangement of the components within the cell membrane.
Because the hydrophilic parts of the components are facing outwards towards the aqueous fluid
Hmm.
because they are polar.
Mm.
So
We
they
can say
have the
simply that whatever the component present in the cell membrane, the amphipathicity of the phospholipid will decide whether that component will be interacted more with the hydrophilic part or more with the hydrophobic part. Okay, the amphipathicity of the phospholipid bilayer decides with which part of the phospholipid bilayer the interaction will be more. Okay, of the incoming
yeah.
component. Like if we can say if the protein, the proteins are also of two types. They are polar as well as there are non-polar proteins. So if there is non-polar protein, so obviously the phospholipid bilayer will tend that that protein should be more in the within the hydrophobic region. If we talk about the non-polar protein, right? And if you have a polar protein, so the phospholipid bilayer will tend that polar protein to be present in the polar part. And if the protein has both the nature, actually it has the nature of the polarity. It has polarity and non-polar. It is polar and non-polar in nature. Most of the proteins. they tend and they act like that. Okay, so they have those two moieties. So it means the polar part, the polar part of the protein will interact with the hydrophilic part of the cell membrane, and the non-polar part will interact with the hydrophobic part. Clear?
Yeah.
Okay. Intrinsic proteins are embedded in the membrane with their arrangement determined by their hydrophilic and hydrophobic interaction regions. Intrinsic proteins may be found in the inner layer. You see what they have mentioned? That the intrinsic proteins, they may be found in the inner layer, the outer layer, or most commonly spanning the whole membrane. Spanning the whole membrane means they can span, like they can present over here. Sometimes they are present over here. Sometimes they are present over here. Okay. Sometimes they are present over here. So this is what they are saying. Okay. The intrinsic proteins, they are present in the inner layer. They may be present in the outer layer, or most commonly they will be spanning the whole molecule, in which case they are known as transmembrane protein. Okay. Now they will be known as transmembrane protein if they span. What is spanning? To move from one place to another
Can you
within the
repeat that?
membrane.
You're repeating.
I am talking about the intrinsic proteins. Intrinsic proteins are those which are embedded in the membrane. Okay, these are embedded in the membrane, and these proteins, they can, now they're saying these proteins can be present within the inner layer. Okay, this is the inner layer, or they can be present in the outer layer. This is the outer layer, or they can span. They can span throughout the membrane, like they can move throughout the cell membrane. These type of proteins, intrinsic proteins. So when they span, in that case they will known as transmembrane proteins. Are you getting this?
Yeah.
When the intrinsic proteins span throughout the membrane, in that situation they are known as transmembrane proteins. Okay. And when we talk about the extrinsic proteins, they are found on the outer or the inner surface. Okay, the extrinsic means outer or the inner surface of the membrane, not within the membrane. Okay, this is the difference. They are embedded. If you talk about intrinsic proteins, they are embedded. They are embedded in the membrane. But when we talk about the extrinsic proteins, they may be present inside over here, or they may be present outside. Okay, these proteins, they will have more interaction over the outside or maybe
The
the inner surface. It's not, you cannot say that they're present in the cell. They are saying that their interaction will be with the outside or maybe with the inside of the cell. This is what they're saying. Such
the
proteins,
extrinsic proteins,
sorry,
they're present. Extrinsic proteins, they're present outside the membrane.
yeah, they will be attached. Obviously, they will be attached over here,
Okay.
or they will be present, or they may be present inside the protein, but they are not intrinsic. Intrinsic proteins are those which will be embedded like this. They will be embedded. You know the meaning of the embedment, just like a button in the shirt, or just like what can which example I can give you for the embedment? Like you dip something in the liquid, okay? That is embedment, a solid thing. So that is embedment. And if you just touch with the surface of the liquid, that will be extrinsic protein. Like extrinsic protein, they touch from the outside. They are present on the outside, but they are interacting from the inside. But they are not present in between. Okay, embedment means in between. So the intrinsic proteins are present in between, within the cell membrane.
Yeah. Okay.
Okay. So the extrinsic proteins, when we talk about them, they are found on the outer or the inner surface of the membrane. These are found on the inner or outer surface of the membrane, and many are bound to intrinsic proteins. Many are bound to, now what they are saying, many will be bounded to the intrinsic. Like sometime this is an intrinsic protein, so they will be bounded over here. Okay, extrinsic
Okay.
proteins. Sometimes they are directly attached to the inner side of the membrane like this, or to the outer side of the membrane, and sometimes they will attach to the protein. Clear?
Yeah.
Some are held in other ways, for example, by binding to the molecule inside or outside the cell, or to the phospholipids. What does it mean? By
What
binding
does
to
it
the
mean?
molecules inside or outside the cell. We have done this. Think about it. Binding to the molecule. They are talking about the conjugated molecules. What are those?
The what?
Conjugated molecules. The conjugated proteins.
Conjugated proteins.
Hmm. I think I should take your test from all the previous chapters, right?
I don't know what that is.
Conjugated. We have done that in the very beginning of the classes, like
The
in
fi-
when I was teaching you the biomolecules. I told you the lipoproteins and glycoproteins.
the glycoproteins, the lipoproteins.
They are what? They are conjugated molecules, isn't it?
What does conjugated mean?
Conjugated means conjugation. The two different molecules are combined. Lipid is combined with the protein. That is why it is known as lipoprotein. Similarly, protein is attached with the carbohydrate, so it will be known as glycoprotein. So this is what they are saying. Sometimes the extrinsic proteins they are bind to different molecules. Like if the protein is binded to the lipid, it will be known as lipoprotein. And sometimes the protein is binded to the carbohydrate, it will be known as glycoprotein.
Oh, yeah, I remember this.
You remember this?
Lip. Yeah.
Yeah, thank God. But I will take your
I
test. Huh?
okay. I realized this last week, but I didn't remember the word conjugated.
Gation. Okay.
Conjugate.
You must know this because they ask the specific terminologies in the biology. Conjugated molecules. Okay. These are conjugated molecules. And these this is what they're saying. The extrinsic proteins they may present in the inside or they may present on the outside